The forensic researcher used DNA analysis to link the suspect to the crime scene and the victim by comparing the genetic profiles extracted from biological samples collected from the suspect, the victim, and the crime scene. DNA analysis is a technique that determines a DNA profile for legal and investigative purposes by looking at short segments of DNA that vary from person to person. The forensic researcher used a method called short tandem repeat (STR) analysis, which is based on the statistical calculation of the rarity of the produced profile within a population. STR analysis uses polymerase chain reaction (PCR) to amplify small amounts of DNA and then measures the number of repeats of a specific DNA sequence at different locations in the genome. The forensic researcher found that the suspect's DNA profile matched the DNA profile obtained from a bloodstain on the victim's clothing and a semen sample recovered from the victim's body. The probability of a random match between the suspect's DNA profile and the crime scene samples was extremely low, indicating a strong evidence of his involvement in the crime. The forensic researcher also excluded other potential suspects by comparing their DNA profiles with the crime scene samples and finding no match. The forensic researcher's findings were presented in court as expert testimony and helped to convict the suspect of murder.